Okay, so this is going to be a little mini lecture on exercise 1 in your enzymes chapter. This exercise is going to investigate catechol oxidase activity, and let's start off by talking about catechol. So catechol is a organic molecule found in some plants. So it's found in the actual vacuole of the cell, plant cell. And when the plant is damaged, let's pretend it's an apple. So you drop an apple on the floor and you cut it open, it's all brown tissue. So when that tissue is damaged, this catechol is released and it mixes with the enzyme catechol oxidase. You know, oxidase is the enzyme because it has that ACE at the end. So it's mixing with oxidase and it creates a product called benzoquinone. And this benzoquinone actually acts as an antiseptic. It's toxic to bacteria, and it prevents the decay of that damaged tissue. So I'm going to pull up the actual reaction so that you guys can all see, and it's also found in your lab manual, so you'll be able to see that. So here we go. We have our catechol and our catechol oxidase mixing with our catechol, and then it basically catalyzes. It removes the electrons and the hydrogens from the catechol, and it's going to create benzoquinone. Okay. So in the actual experiment, we're going to be using potato extract, or I used potato extract, and I had 3 test tubes. So let me draw out those 3 test tubes. Here is test tube number 1, test tube number 2, alright, and test tube, looks more like beakers, but they're test tubes, test tube number 3. Alright, so 3 test tubes, and let's actually pull up what was inside those test tubes. And you can also find this in your lab manual. So let me pull up that table. Done. Move it down here so we have some room. Alright. So in our first test tube, you saw that it had distilled water, catechol, and distilled water again. All of the amounts within each of the 3 test tubes should equal the same at the end, which is why you have that distilled water twice. Test tube 2 has the 5 milliliters as well, and it has the 10 drops of catechol as well. But instead of the distilled water like test tube 1, we actually have that potato extract. Now take a look at test tube 3. We have the distilled water twice, and we have the potato extract, but no catechol. Now there's something called positive and negative controls. Let's think about that. A positive control is going to be a control that gives you a positive result. It's going to give you a result that you know what it's supposed to look like. And a negative control is going to give you a negative result, a result that you know what a negative result is supposed to look like. And then we have our experimental, so what you're really not too sure what's going to happen. Okay? So I did the experiment out, and in the experiment you're supposed to add all the amounts, and what I ended up getting as my result, let me just run over here, and I'm going to crop it just so that you can see just the test tubes. Done. Alright, so these were my results. So this one is test tube 1. Remember, test tube 1 had the distilled water, the catechol, which is colorless, and the distilled water. So that one stayed clear. Test tube number 2, this one had the distilled water, the catechol, again, clear, and the potato extract. So what do you think that one made? Then we had test tube number 3. Test tube number 3 had the 5 milliliters of water, the drops of water again, and the potato extract. Okay? So what I want you guys to do, take a look at this photo, and I would like you to answer questions 2, 3, and 4. I also want you to design a simple table of the results that I got, and then explain your results in terms of a hypothesis that you should have come up with. And then you can answer, like for the discussion, you can answer your results in terms of your hypothesis, and then number 2, what evidence would you support the assumption of the catechol oxidase was the enzyme studied? Okay. If you have any questions on that, let me know, and I'm going to move on to exercise 2.